Today I will talk about graph neural networks in computational biology. And there has been a tremendous surge of interest in leveraging graph neural networks and graph representation learning, in particular, for learning meaningful representations of biology and meaningful representations of data sets and entities we encounter in biology and medicine. And so over the last few years, we have seen several prominent examples where GNNs have been used successfully to learn representations that have enabled critical predictions in a variety of downstream tasks, including predictions about discovering novel interactions between proteins, information about potentially successful disease treatments, opportunities about regarding novel drugs, and so on. And so that has that, of course, is very interesting, and particularly a natural question to ask is, why do we see that graph neural networks achieve such success on biomedical and biological data? And so to answer this question, we need to think a bit about biology and observe that biology is actually highly interconnected with, in the sense that the way drugs work when people take them is not that they work independently of each other, but the way drugs work is that they actually affect certain number of molecules in human cells that are typically proteins. And those proteins interact with each other, and then these effects are then propagated. The way they are propagated in human body and in human cells is through those underlying biological networks. So we can really see that biology is interconnected at various different scales, from the scale of genomic sequences, where we see interactions between parts of the genome, to the scale of interactions between proteins and molecules within a cell, to interactions of external agents that are included in the human body, such as drugs and therapeutics, all the way to the scale of populations or entire ecosystems, where we have, for example, healthcare systems and dependencies between different institutions, different hospitals, or different kinds of symptoms within healthcare records. So the effects of many of biological interventions, many of biological entities are there really network effects. And because of that, it makes lots of sense to study the biology through the lens of networks and interactions between biologically relevant entities. So to unpack this a bit, we can try to understand why are networks in biology so powerful. And I will take a particular example to explain that. So let's take a look at the human protein interaction network. This is a network where nodes represent human proteins, and two proteins are connected by an edge if the proteins physically interact, meaning that at some point in time, the two proteins are physically close in the cell. And you might want to know, well, why are then proteins interacting with each other? If we look at a particular subregion of a protein interaction network, which might be this particular subgraph on 5 human proteins, where we see 5 interactions, is there any significance between these two protein MXS1 and B92? So that in the sense that, what does it mean and what are implications of them interacting with each other? And to answer this question, what we can do is to overlay on that protein interaction network information about what are diseases that those proteins are involved in. And if we do that for this particular example, we can see that these 3 proteins that are highlighted here in red are all associated with the disease, with the same disease, meaning that when there are certain alterations in those 2 proteins, a particular disease would develop. And so what is interesting to explore and observe is that if we overlay information about involvement of proteins in various diseases onto protein interaction network, we can see that proteins associated with the same disease tend to agglomerate or cluster in certain regions, in certain neighborhoods of the network. And that has, over the last 2 decades, yielded to a very powerful paradigms in biology and medicine, because it's powerful as it allows us to identify new proteins that might be involved in disease by simply exploring the surrounding and network neighborhoods of those proteins that were already known to be associated with the disease. That particular long-standing paradigm is known as local hypothesis, In other words, it means that proteins involved in the same disease, proteins that when mutated are lead to the same disease, they have an increasing tendency to interact with each other, meaning that they would be connected in our PPI network. So the implication of that local hypothesis is that mutations in interacting proteins often lead to similar diseases. And now that we know that, we can effectively leverage that information as prior information for our machine learning models. While I explain this particular local hypothesis in the case of human protein interaction networks and diseases, similar findings apply to a broad range of different kinds of networks that we can see in biology, going from protein interaction networks to networks where nodes are patients related and connected based on familial or relationships or similarity of their genomic profiles, to networks representing hierarchies of cellular systems, networks representing disease pathways, gene interaction networks, cell-cell similarity networks, and many others. And so that notion of local hypothesis, where we see that entities with similar properties or tend to cluster and agglomerate in certain network regions, is really one of the core observations and motivation and guiding principle that can explain why graph neural networks are so well suited for the analysis of biological networks and why they have already led to several very successful applications to biomedical problems. So that's all great and fantastic vision that we can share. It's a nice motivation for why it makes sense to use graph neural networks for biological data. But biological networks also present some fundamental challenges that we can solve through algorithmic innovation. So there are a number of challenges. I will just mention 3 here. So the first challenge is that biological networks tend to involve heterogeneous interactions that span from the molecular level all the way to the level of whole populations or societies. And so typically the challenge is how to computationally operationalize the data, how to even construct then graphs that are then amenable to machine learning. The second challenge is that networks in biology contain data that come from a variety of different resources, including experimental readouts, curated annotations, and metadata. And we really need to consider all those data jointly, because no single data type can really capture all the factors that are necessary to understand a particular complex phenomenon, such as a disease. It really is that we need to jointly consider the data from diverse resources to do so. And finally, biological networks are by definition noisy, and they by definition are incomplete because our understanding of the nature and biology is incomplete. And they are noisy because inherent natural variations, as well as limitations of current biotechnological platforms that generate the data, which means that we need to operate with the assumption that our data will be missing, it will contain repeated measurements, even contradictory observations that can plague the analysis, and methods need to be robust against these issues. So the plan for today's lecture is the following. I will talk about 3 vignettes that will highlight some of the challenges for work dealing with biological networks, as well as proposed solutions, and show examples of successful applications of graph neural networks in computational biology. So the parts that I will touch are, first will be the question related to modeling safety of drugs and safety of combinations of drugs. Second, I will talk about predicting patient outcomes and disease classification. And finally, we will discuss methods for identifying effective disease treatments and for identifying promising therapeutic opportunities for emerging diseases. And along the way, I will also highlight some of new technical work that was motivated by these specific biomedical problems. So let's start with the first topic, which is that of modeling drug safety and modeling drug combinations. So this work is really motivated by the problem that is known as polytherapy, sometimes also called polypharmacy. It refers to the situation where patients take multiple drugs at the same time to treat complex or coexisting disease. This is a problem that is very common, actually. 46% of people over 65 years take more than 5 drugs that are prescribed to them by the doctor at the same time. It's not uncommon to encounter patients that take more than 20 drugs to treat heart diseases, depression, cancer, some other complex disease. So the problem with that is that when a patient takes several drugs at the same time, they're at much higher risk for developing adverse events. And there are estimates that around 15% of the US population is affected by those unwanted side effects. And that represents a huge burden, both for individuals as well as for the entire healthcare system in terms of rising healthcare costs. So when I said that individuals are at higher risk of developing unwanted adverse events, what did I actually mean by that? So when patients take multiple drugs at the same time, because we now know the drugs don't act independently of each other, they might give rise to unexpected interactions between drugs. So what does that mean? Let's look at this hypothetical scenario where we have a system with just 2 drugs. We have a blue pill and a red pill. So when a patient takes blue pill alone, there are no side effects observed, and the desired therapeutic effect is achieved, and everything is well. When a patient takes red pill alone, no side effects are observed, and everything is okay. However, when a patient takes blue and red pill together, then with certain probability, the patient would experience side effects when taking that combination of drug. So that is surprising and unexpected, because if the 2 drugs would act completely independently of each other, then we would not expect to see any significant side effects where the 2 drugs are taken together. So our learning task for this part of the talk is the following. We want to design a model that will be able to predict how likely a particular combination of drugs lead to a particular side effect. So that is the task that we want to solve. Okay, so a natural question to ask is, now that we know what is our question, is modeling drug combinations and modeling drug interactions really a challenging problem? And the answer to that is yes, it's a very challenging problem. Why is it so challenging? First, and that's perhaps the core challenge, is that we can see a combinatorial explosion of possible combinations of drugs. Even if we take only a set of drugs that are approved in the US and we consider only combinations that consist of 2 drugs, there are over 13 million possible combinations of 2 drugs. There are over 20 billion possible combinations of 3 drugs and many more higher order drug interactions. So the implication of that is that it is certainly infeasible and to test safety of those combinations of drugs in the lab, which effectively means that these drugs are approved by FDA and other regulatory agencies. They get to the market, and then patients take them, and then we see what happens. Right? So this is a huge opportunity for computational models to prioritize and a priori identify and flag those combinations of drugs that might be unsafe before they are really used in real patients. The second problem is that unexpected drug interactions are by definition non-linear, non-additive effects. Right? An interaction is defined as an effect that is different from what is an additive effect of individual drugs, which presents its own set of challenges. And finally, you might think of this problem as a kind of big data problem, because we'll be working with the set of all drugs in the US and all adverse events associated with those drugs. And there will be millions of those adverse event reports. But very quickly, we get down to the small data problem, where actually for any particular combination of drug, there is just a small subset of patients that take that combination of drugs. And there might be actually no information available about those combinations that are not yet used in patients. And so that's another challenge. So to tackle this particular challenge, what I will mention is an approach that is called Decagon, that uses graph neural networks to predict safety of combinations of drugs. So Decagon was defined on a polypharmacy knowledge graph that had the following structure. It's a graph with 2 kinds of entities, drugs and proteins. So in this particular case, drugs are represented by green triangles. An edge between 2 drugs has a particular type. So an edge of type R1 between 2 drugs would indicate that a specific type of interaction, a specific adverse event, R sub 1, is observed when in patients that take the 2 drugs together, and that side effects cannot be clearly attributed to either drug alone in that pair. Of course, in addition to knowing include information about existing interactions between drugs. We want to understand how those drugs actually work in terms of what part of the body and they affect and how they change activity of human cells. And so for that, the second mode information, the knowledge graph, connects drugs to their protein targets, and that proteins are represented by orange circles. And then we know that proteins interact with each other, which gives rise to this underlying protein-protein interaction network. So given this multimodal polypharmacy network, the approach that I will describe, that is called Decagon, has two components. And so in the first component, in the encoder phase, it will take the multimodal network, it will learn an embedding for every drug and every protein node in that graph, and then it will average those embeddings to predict labeled edges between drug nodes that would correspond to predicted drug interactions or adverse events of combinations of drugs. So let's just briefly take a look at that. So the key idea for generating embeddings of drug and protein nodes in that graph was to explore the local and look at the local network neighborhoods for every node in the graph and separate them by distinct edge types. So that is achieved through two phases, where in the first phase for every node, say node V, we would determine a node's computation graph, and there would be a separate computation graph for each edge type that exists in the local neighborhood of that node V. And in the second phase, the idea is to take those computation graphs and learn what is the best way to propagate neural messages along edges of computation graphs and then non-linearly transform them. And so in this particular case, we see that for node V has two first order neighbors that are of type R3 and three second order neighbors that are of type R3. So that would then define a particular computation graph for node V for an edge type R3 that would specify how information is propagated along edges of that edge type. So this gives rise to multi-relational graph encoder, and in 2018, when the model was developed, the architecture was relatively simple, and I'm sure you're very familiar with these kind of architectures by this far in the course, so I will very briefly describe it. The idea here is that in Decagon, node embeddings were initialized by node features, which in the case of polypharmacy problem were features representing additional information about drugs and proteins, such as drug structure and protein memberships in pathways and so on. So embedding for node V, and then at layer 0 was just initial node feature of that node. And then at each layer of the GNN, the embedding for node V was updated based on this particular equation, which what it did, it looked at all possible edge types that that node V was involved in. And for each neighbor of node V in that particular edge type, for each neighbor U, the method took the embedding of that node U from the previous layer, K-1. It combined that embedding by a trainable weight matrix that was specific for the edge type that we are currently looking at. And sort of that aggregated result was then combined with the embedding for node V from the previous layer and non-linearly transformed to get an updated embedding for node V at layer K. The weights for combining the embedding, its nodes embedding from the previous layer, as well as the embeddings of the neighbors at that point was defined by normalization constant, which was fixed based on the node degrees. Today, what we would want to use are attention mechanisms. But this particular equation was then applied some large K times, where with large K would define the number of layers, and the final embedding of the node is the embedding after K layers of that neighborhood propagation aggregation schema. So that is fairly standard multi-relational graph encoder by now. So the result of that graph encoder were embeddings for drug and protein nodes. And so once embeddings for those nodes were obtained, then the edges were labeled edges between drug nodes were predicted. And for that, the input consists of embeddings of two nodes, C and S. Those would be two particular drugs. And so embeddings of those two nodes, C and S, were combined by a particular type of tensor factorization that was a style of Dedekind tensor factorization that took embeddings for a pair of drug nodes and combined and multiplied them by two diagonal matrices that were relation specific and essentially encoded in a contribution of specific embedding dimensions towards a particular side effect. And then a matrix R, which was a small fully non-zero matrix that allowed the model to capture any kind of dependencies between any pair of dimensions in the learned embedding. And so the output was then of this decoder were then predicted edges for every pair, for every particular side effect. The model would return the probability that the given pair of drugs would interact through that particular side effect. Okay, so in order to then apply this model to the polypharmacy knowledge graph, the first thing to do is to construct the actual polypharmacy knowledge graph. So the data that was used to do so was captured molecular drug and patient data across all drugs prescribed in the US. And for that, a unique data set was built that consisted of over 4 million drug edges. Those are currently known interactions between pairs and drugs that were retrieved from the FDA, information on drug protein edges and information on protein edges, as well as side features on nodes. So that gave rise exactly to this multimodal knowledge graph that we have seen earlier, which had over around 5 million edges altogether, and it was highly multi-relational as there were 1,000 different edge types in that graph. Once the data set was constructed and Decagon was fully specified, then the Decagon model was trained on the polypharmacy knowledge graph in an effort to answer questions of the following form. So the users could then pose a query, for example, whether a form of statin and ciprofloxacin, which are two drugs, when taken together, would they lead to breakdown of muscle tissue? That effectively meant that in the back end, the model was asked with predicting the probability that these two drugs are connected in the graph by an edge of type R2, if R2 would indicate breakdown of muscle tissue. So once the model was trained, it was compared against several baseline algorithms at that point in time. So the results are shown in this slide, where on the y-axis we see performance, higher values indicate better performance as measured by AUROC and average precision at top 50. And the performance of Decagon model is shown in red. Performance of other baselines is shown in brown, blue, and green color. So the particular baselines that were considered here was a simple risk called tensor factorization, which can be thought of as Decagon that does not use the deep graph encoder. And so that test has shown that it's really important to use graph convolutions to learn powerful embeddings. So then Decagon was compared to a multi-relational factorization, which is and it's the relative improvement of Decagon over that factorization showed that it's really important to be able to model side effects jointly because there are some correlations between side effects. And finally, then Decagon was compared to a shallow network embedding model, which was an Autovik style model. And the improvement there showed that the message passing network is much better in terms of performance in this particular application than some of the random walk based embeddings at the time. So another test that was done was to apply Decagon in a temporal fashion, meaning the Decagon was trained on all the data generated prior to 2012. And then the model was frozen and it was asked to make predictions that the model is most confident about. And we then tested, well, how many of those predictions have been confirmed after 2012? So what is shown in this particular slide is our top 10 predictions made by the model. And so recall that each prediction comes in the form of a pair of drugs and a side effects associated with that pair of drugs. And so for 5 out of those top 10 predictions, direct evidence was found, published after 2012, showing examples of real patients who were on a particular combination of drugs and have experienced the kind of side effect that Decagon has actually predicted will happen. So for example, for drug prediction number 8, there are several reports of patients who took a statin and amlodipine together and experienced muscle inflammation. And those reports were made after 2012, and which provides evidence for prediction made by the model. Some of the novel predictions in this space are novel hypotheses that warrant further analysis and downstream investigations in several collaborations with domain experts who work in these disease and drug areas that has actually been done. A more recent follow-up on the Decagon study is concerned with modeling adverse events at the level of patient groups in an effort to make predictions about what are adverse events that might be associated with certain group of individuals, where those groups can be defined by age, by gender, or by individuals that have certain diseases. And so what I'm mentioning this because the data is already pre-processed and provided in this study. And so those who are interested in potentially leveraging GNNs for doing more personalized level adverse event prediction, this is certainly something that is possible now to do with this data set that contains over 10 million adverse event reports. Okay, so this concludes my first vignette, or first a large topic for today. And the next topic will change the gears, and we will move away from predicting safety of drugs. Instead, now we will think about diagnosing patients, which is an important, crucial question that first need to be answered before any drugs can be actually, or treatments can be actually identified and recommended to the patient. So let me start with the motivation of disease diagnosis. The core information for a disease, for diagnosing a disease, broadly speaking, is the information about phenotypes that are seen and observed in a particular patient. So phenotypes can be defined as some observable characteristics that can be seen in a patient, and they result from various interactions between the genotype and the genomic information within the patient, as well as environment in which the patient lives. So physicians typically utilize various standardized vocabularies of phenotypes to describe human diseases. For example, a very prominent standardized vocabulary of phenotypes is known as human phenotype ontology, and that codifies a variety of different phenotypes and how they map to human diseases. So by modeling diseases then as collections of phenotypes or that represent the disease, we could then diagnose patients based on what are the phenotypes that they have, based on what are the symptoms that those patients would have. So in machine learning, we can define the diagnosis task then in the following manner. We will start with a graph. So consider, in this particular example, a graph that is built from the standardized vocabulary of phenotypes. That would be a graph where nodes are phenotypes, that those might be visible symptoms, as well as various phenotypes at the molecular or genomic level. And edges between those phenotypic nodes would indicate different kinds of relationships between phenotypes. We can, in that setting, the disease would then be represented as a set of nodes or as a subgraph in that phenotypic network. So the learning task would then be, the diagnosis task is then concerned with predicting the label or predicting the disease that is most consistent with a particular subgraph S that describes the patient. So you can think of now that patients are these different subgraphs of phenotypes that patients have, and the goal is to predict labels, which in this particular case are colors of these subgraphs, and those labels are the diseases that are candidate diseases patients might have. Okay, so given this formulation, let's discuss the algorithm for this problem a bit, and then we will return back to the data and the diagnosis task. So in the context of graph neural network, our problem formulation is the following. The goal here will be actually to learn embeddings for subgraphs such that those embeddings for subgraphs that are learned, we capture well the topology of and interactions between nodes, as well as where they exist in the context of the underlying graph. So that means that that the input, which then consists of some underlying graph G and a large number of subgraphs that have labels. And the goal is to learn subgraph embeddings such that two subgraphs S sub I and S sub J that have similar subgraph topology, and I will define next what that means, they should be embedded close together in the embedding space. So represented by nearby points in the embedding space that we want to learn. So why are subgraphs challenging? So why can we, for example, just take our favorite node level embedder, and by now I'm sure you have heard of many node level embedders in the course, and essentially just learn node embeddings and take an average of those node embeddings, or somehow aggregate those node embeddings to arrive at a subgraph embedding. So there are several reasons why we might lose a lot of information if you do so. So one of them is that the subgraphs that we would encounter in the diagnosis task are subgraphs of varying sizes. And then the question is really how to represent those subgraphs effectively that are not simple k-hop neighborhoods around a single center node. Second, subgraphs would have rich connectivity structures, both internally as well as externally through interactions with the rest of the graph. So how can we inject that information into our GNNs? And finally, subgraphs that are given by sets of phenotypes for patients can be localized in the sense that they would reside in a single region, in a single large neighborhood of the graph, such as this hypothetical example where the subgraph is indicated by this red blob that is core to the center of core of the graph, or subgraphs may be distributed across many multiple local neighborhoods. And so then a simple averaging of node embeddings might not be an effective strategy. So to tackle this problem, what was developed was a subgraph neural network model. And so the problem that the subgraph neural network is solving is the following. It assumes that given some set of subgraphs S, this set has n subgraphs, S1, S2, up to Sn. SubGNN specifies then a neural message passing architecture that will generate D sub S dimensional subgraph representation for every subgraph in that set. And it will use those representations to learn a subgraph level classifier. So a function that will then map those subgraphs to some number of discrete labels. And the way it will do so is by really taking into account different kinds of subgraph topology. In particular, the parts of subgraph topology that we are interested in is related to capturing the connectivity within the immediate local neighborhood of each subgraph, both at the internal within a subgraph, as well as at the border of the subgraph and the graph G. The information about the structure of the subgraph in the sense of are there certain motifs that are over or underrepresented in a subgraph, as well as where is that subgraph located relative to the graph G in which it exists. So a quick note on problem formulation that I want to give here is that SubGNN puts forward a definition of subgraph prediction learning task. This is different from other canonical tasks on graphs that are primarily concerned with node level predictions and representations, where the goal is to predict properties of an individual node in the graph, or link prediction, which is concerned with predicting properties of a pair of nodes, or graph level prediction, which is concerned with predicting properties of entire graphs. So SubGNN is concerned with predicting properties of subgraphs of non-trivial size, meaning not subgraphs of size just 2, which would correspond to an edge. The way this is done is that SubGNN consists of 2 parts. So the first important part is a message passing scheme that is hierarchical in nature, and it specifies the way neural messages are propagated from anchor patches, which are helper graphs sampled from G, to subgraphs S that we want to embed, and how those messages are then embedded, aggregated to arrive at the final subgraph embedding. And in part 2, that routing of messages is really done to 3 distinct channels, where each channel corresponds to a distinct aspect of subgraph topology, position, neighborhood, and structure that we want to capture. capture. Very briefly, let me describe these two parts. So the subgraph message passing scheme has the following structure. There are neural messages specified. Those messages are specific for each of the three properties. So they're denoted M sub X, and those messages are propagated from anchor patches to subgraph components. Anchor patches are just helper subgraphs that are sampled randomly from the underlying graph G. And there are three different kinds of patches depending to capture position, neighborhood, and structure. So then the message passing is then formulated as the message from this anchor patch A to subgraph S is sent, and it's scored based on its weight. Its contribution is weighted by a similarity function between a subgraph component and an anchor patch that represents a weight of how much an embedding of the anchor patch will contribute to embedding of the subgraph. So this is visually shown here in the right part of the slide, where the subgraph that is part of the data that we want to embed is S sub 1. This subgraph actually has two disconnected components. One is here on the left part, and the right part of the right extreme of the graph. And in the message passing phase, those helper patches, anchor patches, which are in gray, send their messages to the subgraph component. Those set of messages that are received at the subgraph component are aggregated, and then the aggregated message of the subgraph component is combined with its previous component, with its previous embedding, to get a property-specific representation of subgraph component. So H sub XC is then an embedding for subgraph component C. So it would be just for this one component of the subgraph S sub 1 that captures the aspect X, which can be position, neighborhood, or structure. So the way this message passing is done in three separate channels, and each channel describes is designed to capture one aspect of topology, position, neighborhood, and structure. Each channel X has three key elements. Those elements are similarity function that weighs messages exchanged between anchor patches and subgraph components. Then an anchor patch sampling function that specifies how these helper patches need to be sampled from the underlying graph in order to capture neighborhood or see and capture diverse neighborhoods and diverse structure and diverse positions. And finally, how to encode those anchor patches. And these functions can be learned or predefined. And so the recap of that is that we can think of this problem as the problem of learning subgraph representations as a message passing network that has three distinct channels, and those channels output channel specific embeddings that are then aggregated to produce a final subgraph representation Z sub S for subgraph S. So let's now return to our problem of disease diagnosis. And the first thing we will do, we will test whether that method works on some simulated synthetic data sets to really see whether the method can truly capture the distinct aspects of subgraph topology. And the setup for that was the following. In each case, each data set started with the base graph. And base graph was then sprinkled with a large number of subgraphs. There were two strategies to define subgraphs. One strategy was the planting strategy, where a subgraph was generated and it was planted on top of the base graph, or it would be essentially just joined in the form of, in similar way as it would be stapled together with the base graph. In this particular subgraph, in this particular synthetic data sets, for each subgraph, a label was defined. And so labels were defined based on certain network structural properties of those subgraphs. So one particular synthetic data set was called density, where that was a data set where that consists of the underlying base graph and then a large number of subgraphs. The labels of those subgraphs were binned values of network density metric. And similar data sets were constructed based on cut ratio metric, coreness, and component. Why would you want to do that? The idea was to do that to test whether really the most informative channels for certain types of labels are those channels that really truly determine the label. So this is really a controlled environment of testing performance of the model. And so those results show that SubGNN is an effective strategy for learning subgraph embeddings that outperforms various simple ad hoc measures that one could think of. For example, taking a simple average of node embeddings and obtain subgraph embeddings in this way, or introduce a meta artificial node and then embed those meta nodes that could serve as proxies for subgraph embeddings, or even doing graph level embeddings. Finally, these results also show that the SubGNN can capture well different aspects of subgraph topology. That can be studied by looking which of the three channels, neighborhood, position, or structure, would be most informative for predicting certain kinds of labels. So for the case of density, the label would primarily be determined by the neighborhood of the subgraphs. So what was then tested is whether the neighborhood channel is most informative for predicting the density labels. And indeed, that was the case. Okay, so now that we know that SubGNN works well, let's return to our real-world data set. So there were 4 real-world data sets that were designed for the problem of clinical diagnostic tasks. So each of the 4 data sets consists of a base graph and subgraphs with associated labels. So the two data sets that I want to bring your attention to are called HPO Meetup and HPO Neuro. So these are the data sets where the base graph is the graph of from the human phenotype ontology. So this is a graph where nodes are human phenotypes, edges are hierarchical relationships between phenotypes and symptoms that we see in human populations. And then labels are defined by certain, then subgraphs are defined by subgraphs of metabolic diseases and metabolic disorders that affect humans. And in the HPO Neuro data set, then subgraphs represent various neurodegenerative diseases. So the goal of these two particular data sets is to predict subgraph labels that correspond to various metabolic or neurological diseases that are consistent with the underlying phenotypes. So this simulates the environment of diagnosing patients that might have metabolic or neurological disorders. So let's see performance of SubGNN across those 4 data sets with a particular focus on the human phenotype ontology neuro and metabolic data set. And so what we can see is that the performance of SubGNN when implemented with when compared to baseline models is substantially better, and SubGNN outperforms those baseline models considered here by up to 125%, which is a really incredible improvement in performance, which clearly motivates the use of subgraphs rather than simply considering node level or graph level aggregation. Okay. So this concludes the second part of this talk, which was concerned with using the power of subgraph embeddings and GNNs for disease diagnosis tasks. And in the third part of the talk, I will focus on an important endpoint for drug development, which is that of drug efficacy. So in the first part, we talked about drug safety, but drug safety is one of 2 corner endpoints that we care about in the drug development process. In addition to drug safety, the second important endpoint is efficacy, where the question is to understand whether a certain molecule that might be candidate drug will really have positive therapeutic effect in patients. So let's start with this third part of the talk. The motivating problem for this task was the question of how to find cures for emerging diseases. This particular work was motivating by the onset of the COVID pandemic last year, where it became clear that at the beginning of the pandemic, the need was for rapid deployment and identification of drugs that might have positive therapeutic effect in in COVID-19 patients, but simply traditional approaches of iterative development, experimental testing, and clinical validation and approval of new drugs were not feasible because they on average take over a decade to bring and design a new drug from scratch and bring it to market. And that was simply not feasible. So a more realistic strategy relied on repurposing of drugs, requiring us to essentially identify what are drugs that are clinically approved that might have therapeutic effect in COVID-19 patients. And so the particular study that I will be mentioning is the one that was conducted, that started at the beginning of March last year. And the most parts of it were done between March and last summer. And it really demonstrates an opportunity of how, in this particular case, graph neural networks were able to compress years of work into actually weeks, in some cases, actually days of work, through very close collaboration between machine learning models, predictions that provided by those models, and end users, which in this case were virologists and experimental biologists who took predictions and experimentally screened them in the lab. Okay, so that is our goal for this part of the talk. Let's now try to unpack and define the problem of drug repurposing first, and then describe our technical approach for this problem. So as I mentioned, the discovery of a new and development of new drug from scratch is a very lengthy and resource-intensive process. It on average takes more than a decade, and it costs $1 to $2 billion to design a new drug from scratch. And so faced with these skyrocketing costs for developing new drugs, researchers are looking at ways to repurpose older ones, to essentially take drugs that are already on the market and ask, are there any other kind of diseases that those drugs might be effective in? And that is a very appealing strategy because it would be much shorter and potentially much more effective. A most famous example of a drug that was repurposed on the market is Viagra, that was initially designed for treating cardiovascular diseases. But then during clinical trials, it turned out that there might be other indications that it would be useful for. And so while in the past, those repurposing strategies were primarily coincidental findings, there is now a concerted effort of, can we systematically go through drugs that are clinically approved and are on the market and identify possible therapeutic opportunities? So the core of this question, or the core of this problem, is the following question, which we can think of it as a link prediction problem in a bar pie chart graph, where on the left we have a set of drugs. Those might be all approved drugs plus investigational experimental drugs, some drugs that failed in clinical trials, but otherwise are not hugely unsafe for patients. And on the right, we would have tens of thousands of diseases, which are codified diseases that could be described, for example, through a set of phenotypes that we were discussing in the second part of the talk. So edges connecting drugs to diseases here would indicate known treatment relationships. The problem that we have here, our goal is to have a model that will predict what diseases a new molecule or an existing drug might treat. So is this a challenging problem? Why is finding treatments for a new disease in particular such a challenging problem? So when we are concerned with the question of finding treatment for a new emerging disease, that means that we want to identify drugs that might treat the disease, but there are no currently known drugs for that disease. So, and COVID was a very, very prominent example of that early last year. And so technically that means that that's not surprising because we know in machine learning that generalizing to new phenomena is typically hard. Prevailing graph neural network methods still assume and require abundant label information in order to be really trained effectively and achieve high accuracy. However, I would argue that one of the definitive factors of a frontier of biology and medicine are problems where labeled examples are incredibly scarce. And this includes emerging diseases, such as COVID-19, rare diseases, hard to diagnose patients, modeling novel drugs in development, and many, many other problems. So what prevailing methods assume is that we start with graphs that are already richly labeled. That assumption is heavily violated in most exciting problems, where in reality, what happens in the world that we only have a handful of labels for a particular class, if any at all. So for the problem of drug repurposing, and the method and the GNN methodology that I will describe, it really relies on few-shot learning and meta-learning. So I have 2 slides now with just a very brief high-level overview of that, of what meta-learning is, and I assume that many have already heard about this in other courses. So meta-learning is a very effective strategy for learning in the cases where there are a very few small number of labeled examples available for tasks. And so typically the idea of meta-learning strategies is the following. We assume that we train a meta-learning model over a variety of learning tasks, and that model is optimized to achieve the best performance on a distribution of tasks, including potentially unseen tasks. The way to think of meta-learning is that think of each learning task being defined and associated with a small data set, D, that contains both data instances, feature vectors, and some true labels. And so meta-learning is then concerned with finding the optimal parameters, theta star, for the model that would minimize the loss function across those large number of data sets, each describing a different task. So if those models can be found if those parameters theta star can be found successfully, that indicates that the model was able to really successfully generalize from one task to the other, from one data set to the other. And so if it can do that effectively through the process known as meta training, then we can hope that also in the case of at test time, it will be able to very quickly, very effectively adapt to a new task. In our case, that will be a new disease, COVID-19. So this looks very similar to just normal regular machine learning tasks, but you can think of essentially one data sample is not a single node, it's not a single edge in the graph, but essentially a data sample is a small data set, which in our case would be a small part of the graph on which the model is trained. So a second slide of the background of meta-learning is about few-shot learning, which is just a specific instantiation of meta-learning in the field of supervised learning. And I'm mentioning that just because I want to introduce one notation, and that notation is describes the difficulty of few-shot learning tasks, where we would say that we are interested in solving a k-shot n-class classification problem, which meaning that we want to solve a task for that in a problem for which we only have k-labeled examples for each of n classes. So what is shown here in this slide is an example of a 2-shot 3-way image classification, where we would have where each task would consist of classifying images into 3 classes. That's why it's 3-way image classification. And for each class, we have 2 examples. And so the process here would be that during training phase, we sample and define a large number of training tasks. Each of them is an instance of 2-shot 3-way image classification. And so if the model can be successfully primed to solve this to solve this task during meta training phase, then during meta testing time, then the model would quickly adapt to completely new set of classes for which only a handful of labels are given as well. The goal that we will have here, now that we have this background on few-shot learning and meta learning, is how to make predictions on a new graph or a new label set. So essentially, we want to use few-shot learning not for image classification, but we want to use it for graphs. Okay. So the problem formulation of this problem is the following, we are interested in designing a meta-learner. That meta-learner will be concerned with, will need to be able to classify an unseen label set, so that could be a blue label set, by observing some other label sets in the same graph. So during training, the model will be learned only on yellow labels, and we would expect that the model would be able to generalize to blue labels that are defined within the same graph during test time. And so the method that does that is called GMeta. The overview of GMeta, if I present it in similar way as I have done for few-shot learning just a few slides ago, it's very similar to the general few-shot learning setting, but now we are not concerned with solving image classification tasks, but we are concerned with solving graph learning problems. For the case of this explanation, those would be node classification problems, where during training time, we would define a large number of tasks, each from a particular label set. And at test time, the new task will be presented on label set 3 that would not be seen, that was not present during training time. So the key idea that GMeta uses to solve this, to solve the few-shot learning problem on graphs, is that it will specify a signature function for each learning task. That signature function will be defined based on the structure of subgraphs that encompass labeled examples in each task. And then those subgraph signature functions will essentially learn how to map the structure of sampled subgraphs representing task and effectively serve as initialization for a GNN. And so this strategy can be really effectively the strategy of extracting subgraphs that enclose labeled examples and then applying GNN to each subgraph individually is effective strategy for doing few-shot learning. A natural question to ask now is just conceptually, which is an interesting question, what is the value of these local subgraphs that we are using now to transfer knowledge from one task to the other? And the reasoning for why subgraphs are useful here is the following. You can think of there are two fundamental core sources of information that the GNNs are using. One is based on label propagation, where we can think of nodes that have the same label. They tend to be nearby in the graphs. We have seen at the beginning of the lecture today that proteins that tend to interact, they also tend to be affiliated with similar phenotypes and lead to similar diseases. The other core source of GNN power is structural similarity, where nodes that have the same label, they might not be nearby in the graph, but they might have similar network shapes in their local neighborhoods. Okay, so now that we know that in the case of few-shot learning, where labels are really scarce, then we cannot rely much on label propagation. It's simply not sufficient. When only a handful of nodes are labeled, it's very challenging to effectively propagate labels through the entire graph. So instead, we can much better leverage structural information and structural equivalence, which is exactly what these local subgraphs capture as a mechanism for transferring knowledge across tasks. And it is actually possible to show theoretically that local subgraphs that are defined around labeled examples capture much of relevant information for prediction. And beyond theoretical findings, it has been well demonstrated by many studies in the context of biological networks that typically most useful information that describes a particular task in the context of biological networks exist in some 2-3 hop neighborhoods around target nodes. So the use of local subgraphs is then highly motivated. Okay, so let's return now finally to our COVID-19 repurposing problem. And so I will start with description of the data set on which this transfer, this few-shot learning strategy that I described was applied. So the repurposing data set had 3 major components. The first was information on how to even represent COVID-19. Okay, how can we represent it in the form of a graph or some structure that is machine readable that we can actually compute over? And so to do that, we relied on the study that was published in last year in Nature by Gordon et al. And colleagues from UCSF. And what they have done, they have expressed 26 out of 29 proteins in SARS-CoV-2 virus. And they have checked what are human proteins that those viral proteins attack. And so in doing so, they identified 332 human proteins to which the SARS-CoV-2 virus binds. And so that can be used as essentially computable representation of COVID-19. COVID-19 can be then represented by that set or subgraph or structure of 332 human proteins that are directly attacked by the virus. So these are known as viral human protein interactions because there are actually interactions between the virus and humans. The second important information was that on what are other proteins that those attacked proteins interact with. So that is the human interactome network or the human protein-protein interaction network. And the third piece of information was the information about existing drugs on the market. So for each and every existing drug, we pulled information on its known targets, on proteins that those drugs bind to, and that gave rise to those drug modules, which would be a set of proteins for each drug that are targeted when the patient takes the drug. So COVID-19 ultimately was represented as a network neighborhood of human PPI network targeted by SARS-CoV-2 virus. And the few-shot learning strategy was then applied to learn the embedding for COVID-19. That was the new task that we wanted to rank drugs for based on how promising they might be for COVID-19. The way the model was optimized, it was optimized based on known drug disease indications for other drugs and other diseases that are much better understood than COVID-19. So that process yielded an embedding space for approved drugs and diseases. One point in that embedding space was the point that represented COVID-19. So an effective way to identify repurposing opportunities for COVID-19 was to look at what are drugs that are embedded close to the COVID-19 embedding, and then prioritize drugs based on that closeness between COVID-19 embedding and the drug embeddings. And that gives rise to a ranked list of drugs where those at the top represent most promising most promising opportunities for repurposing in the context of COVID-19. The first test to do when you have such a model is to try to get some understanding into how accurate those predictions are. So what this plot is showing, it is an AUROC plot that shows on the x-axis false positive rate, on the y-axis true positive rate of a prediction problem, which was defined as a question of can these models effectively identify those drugs that were in April 2020 investigated on clinical trials for COVID-19. And so a number of baseline methods were tested for this question. The 3 GNN-based methods, the 4 GNN-based methods are called A1, are denoted A1 up to A4, and they showed substantial improvement in performance relative to some other networks medicine-based strategies based on diffusions, random walks, network proximity, and others that are more commonly deployed in the field of drug repurposing. That finding by itself, however, while promising, it's not particularly exciting because the fact that a drug is investigated in clinical trial for COVID-19 does not really mean that it's successful. And so a real test of the accuracy and the ability of the predictions being useful is to experimentally validate predictions. So for that, biologists and virologists from the National Emerging Infectious Disease Laboratories. So this is a national laboratory that can work with live viruses. And it looks like this. So it looks like very kind of almost like some kind of environment in a space where nobody can enter except those with very particular access. And they're able to work with live viruses there. So the virologists there took the top predictions made by the model and then screen those predictions for their efficacy in first African monkey cells, which are Vero E6 cells, a very prominent cell line for COVID-19 tests, and later in mouse, in human cells. So the result of that was that 77 drugs that were predicted showed strong or weak effect, and they were active over a broad range of concentrations. Importantly, those predictions that were obtained by GNN and actually an ensemble of methods, including some of the other network medicine methods, gave an order of magnitude higher hit rate among top predictions than prior work. So prior work would be just brute force tests of all drugs in the lab. And if that is not done in a brute force manner, but it's rather informed by prioritized list of drugs produced by ML models, then an order of magnitude better hit rate is something that we could expect. Since we are in the course which is concerned about networks, there is an interesting finding that I want to mention here. And so a natural question to ask is, great, so this analysis identified 77 drugs. Is there something common to those drugs that inhibited viral growth? And so the answer to that question is yes. So 76 out of 77 drugs that were predicted and experimentally showed that they successfully reduce viral infection were the kind of drugs that do not directly bind to those human proteins that the virus directly attacks. Instead, those drugs rely on network-based action. Those drugs, such as D3 here, would target some human proteins that are not directly attacked by the virus. And those human proteins in turn would interact with some other proteins who in turn, finally, would be exactly those proteins that the virus attacks. Why am I mentioning that is because these kind of drugs, which we now call network drugs, could not be identified by traditional strategies for drug repurposing, which in computational biology and computational pharmacology are based on docking. Okay, so since I'm already running over time, I will wrap up here. So to summarize the lecture today, we talked about interesting applications of graph neural networks in computational biology, and we touched three topics. We talked about using GNNs for modeling in drug combinations and identifying adverse events and concerns regarding safety of drugs. We then talked about opportunities that GNNs present for defining diagnostic tasks and classifying diseases. And finally, we talk about ways for using GNNs to identify promising therapeutic opportunities with a particular angle, which was that of opportunities for never before seen emerging diseases. So this concludes my last lecture here, and I'm very happy to take questions. Thank you.
Thank you very much, Marinka. This was awesome. Would you want to comment a bit more about what do you think are big challenges in biomedicine that are ready to be attacked by machine learning AI methods?
Wow, that's a very broad question. So the way I would think of biomedicine is in the following way. So we can broadly think about certain core problems. So one core problem is related to diagnosing patients. Before any kind of treatment can be identified or any kind of drug can be recommended, we first need to know what is the disease that the patient has. So a core challenging problem that is ready to be tackled from the ML standpoint here is in particularly we need methods for rapid diagnosis of patients, in particular those patients that are hard to diagnose and have many rare novel diseases. You might say, well, that might not be lots of patients, but it turns out that 20% of all patients in the US have this kind of diseases. And on average, patients with rare diseases need to visit 7 specialists before they are correctly diagnosed. That's, again, a huge problem for healthcare systems, for individuals, et cetera, et cetera. So now the data sets are available to expedite the diagnosis tasks considerably. So I think that's one exciting problem and direction to pursue.
Sorry, just to ask a follow-up. So do you think of this as an active learning type problem where you also recommend what other tests to take so that you can diagnose the disease?
Probably not, because the problem is that if you look at those patients, they already went through many, many tests. They they took lots of different tests, and we don't know what else to do. So it's not that much a question of identifying what lab tests should they conduct, but the problem is that we can ask ourselves why we cannot diagnose patients easily. The problem is that they are conflicting in some way, meaning that a patient would come and would present with phenotypes and symptoms that don't match with any known disease classification, right? So the way patient comes to the physician has a huge book they open and they say, okay, disease A, these are the symptoms that I would expect for disease A. And they compare that with patient symptoms, there's no match. Okay, so no disease A. Let's go to disease B. Let's go to disease C. And the problem that it's hard to diagnose those patients is that they somehow have a mixture of symptoms that don't align well with any of the disease. So it's really more about essentially diagnosing them. And the question is, what is a good diagnosis? Probably it's not just saying a patient has disease A, but it has this set of diseases which, or can we learn some kind of distribution over known diseases that capture that patient well? Mm-hmm. And so that's just the first task of identifying what's wrong with the individual, right? The immediate next question is, can one identify successful treatments? And so here, all the questions related to drug discovery and development apply in the sense of, can we repurpose existing drugs? Can we identify combinations of drugs that might work for a patient? Can we do that in a more personalized way? And it's possible now to meaning that the data sets are ready and available to move away from the one-size-fits-all treatment recommendation, which is still more or less the kind of work that I've been describing in the lecture today. It's essentially one-size-fits-all treatment recommendation, but it's now possible to go to a more precise level, at least at the stage of patient groups, meaning what would work for certain genders, certain ages, certain combinations of gender, age, or molecular markers. I think then there is another large group of very important, high impactful questions in biomedicine to ask, which are related to temporal aspects of how diseases progress and how to monitor the patients and help them improve their self-care. So those are important questions to tackle, for which data sets are available as well. And then close collaborations with healthcare systems would offer lots of opportunities for kind of tedious work. Tedious work would be optimizing clinical workflows, identifying medical errors, in proposing systems that have and that use active learning to then identify when to add another drug to a patient, remove a drug from the patient, when to call the patient to come to the clinic. So those are all exciting questions, and some new questions have emerged during the pandemic. An example of a question that is new is that of remote medical visits. That is particularly very exciting topic nowadays in the interface of AI and medical space. In particular, the question is, can we design various AI systems that could help doctors to perform visits remotely, in the sense that one would have it would be possible to take an image a patient could take an image with their app, and that image would be analyzed automatically on the patient side and then communicated to the physician. And here in particular, active learning strategy is most important to, for example, identify what part of the data is most crucial to have in order to make diagnosis, right? And then, can that part of the data be acquired without the patient necessarily visiting the hospital? And if that can be done, that would enable remote diagnosis and management.
This is super cool. So I guess a follow-up question is, especially for the first few problems that you were mentioning, why are graphs essentials? Why are these graph problems and not natural language problems, or I don't know, computer vision problems? Why do you think graphs are essential in this case?
That's a great question. I try to shed some light into this question at the very beginning of the talk, a very beginning of this lecture. And so the answer to your question would have two parts. The first part is that it's very easy to see that most of the problems in biology are codependent, or in the sense of whatever task we pick up, typically there are substantial relationships between important entities in that task that need to be modeled, and decades of kind of fundamental biological research has shown that those dependencies are really important and cannot be ignored. So the relational structure of the data set is very universal. So we have seen that for essentially all three problems discussed in this talk, that that's indeed the case. And it has been shown over and over again experimentally, from protein interaction network, CRISPR gene editing, disease diagnosis task, and so on. So these relations are an important component of the data set. So we would need to leverage models that use that. Second is not only that these relations exist, but they are in most cases really informative, sometimes even more informative than structural data, for example, for the problems of predicting proteins structure. The recent models put out by DeepMind actually leverage relational information about proteins to infer what their structure is about. And so not only the data sets and problems have strong relational structure, but that relational structure correlates well with predictive tasks that we want to produce. And so then some form of graph-based machine learning seems very suitable.
Thank you so much. One more question is about how effective has it been to get the medical community to adapt these methods? That I guess is the question.
So that's a great question, and the short answer to that would be it's challenging. So first, to answer that, by now we have seen several examples of successful deployments of these ML algorithms in general in clinical settings as well as in large pharma companies. So that is primarily true so far for image-based models. There aren't many, or I am not aware of really successful graph-based machine learning model that is applied and used routinely in clinical context or in at least in public clinical settings. Where we are currently at this stage is that typically individual researchers engage with collaborators who would be physicians or clinical researchers and so on, and they would work hand in hand on their particular problems. And one could think of that as validations, experimental validation of predictions. But currently we are not there yet where these methods would be routinely used, where graph neural networks or graph ML would be routinely used in real world. Why is that the case? So I would argue that the main gap that is significant that exists between, say, models published in NeurIPS papers and then these models being deployed in real world is this lag that typically when we apply these models to data sets, they don't generate predictions that users would think of as actionable predictions. What that means is that it's not enough to simply predict whether a drug will treat a disease. It's very hard to convince somebody that will follow up with very expensive downstream experiments in the lab by this high-level point-wise based prediction. So what I didn't discuss in the context of COVID repurposing project, it was incredibly important to essentially provide explanations, allow users to provide feedback to the model, and essentially build some trust and confidence in the model. And so that has now given rise to this umbrella term of trustworthy machine learning, which I think has some bearing in the context of biomedical applications.
Thank you. Then two more quick questions. Could you elaborate on the node feature engineering for drug-drug interaction, the drug combination in the first part of your talk? And then we have one more.
Yes, so for node features in the first part of the talk, we had node features for drugs and for proteins. Node features for drugs were smiles, fingerprints of the drugs, and they were not manually engineered by us, but this is a very common descriptor of drug structure that is used in chemoinformatics. It was retrieved from a database called DrugBank. For protein nodes, the node features were binary vectors that contained information about what molecular pathways those proteins are involved in. For example, if we know that a protein A participates in cell apoptosis pathway. This is a pathway that's incredibly important because it basically tells it has a control over when a cell should die or stay alive, right? So if a protein is a member of that pathway, then there would be a simple indicator 1 at that, in particular, in the relevant dimension of the protein feature vector.
Thank you. And then next question is about the G-meta learning. How somebody is interested, how does it differ from Bayesian network learning and EM algorithms? Could you discuss pros and cons?
Oh, I see. Interesting. So we actually didn't consider any Bayesian network learning method for the problem of G-meta. We compared it primarily with current work on that is graph neural network based for few-shot learning. I can clearly see from this question that it might be interesting to use some Bayesian model and then learn a distribution of drug effectiveness across diseases and then use that to sample a new disease that would approximate COVID-19. The pros of that would be that using the Bayesian models would perhaps give us a better ranking of drugs, because ranking of drugs for that problem was determined on predicted scores returned by GNNs, and we know and returned by G-meta in particular. From several applications, we know that often the scores that are returned by the model are not necessarily well calibrated or indicative of probabilities. So I could imagine that the Bayesian network model might work better for that. So that would be the pros. The cons of that would be that what we found in the context of COVID repurposing is that the G-meta or graph neural network-based models, when implemented with very recent neighborhood sampling strategies from like cluster GCN or GraphSaint, for example, and other, they can scale very well to very large data sets, including, for example, one data set where we looked at information not only for human, but from our 1,800 other species and tried to translate information from other species and model organisms like zebrafish and monkeys to human. And at least in my experience, the GNN models scale better to larger data set than some Bayesian models.
Thank you so much, Marinka, for the excellent lecture, and thank you everyone for asking great questions. Appreciate it a lot. Thank you so much, and all the best to Boston.
Great. Thank you for inviting me.
Yeah, thank you. Bye-bye.
Bye.
Bye.